Thank you so much. I'm also super happy to be here and had the chance to meet all of you again and to see this really nice place here. So I will talk today about tree mortality and to really not totally, only talk about the network and so on, but really to attach to all the great physiology we have heard today and particularly now in this afternoon session. I will start with talking about mortality mechanisms. They are all related to drought and heat stress. So most of you probably have seen this publication out by Mantova, and there's this idea of this tree mortality spiral. So you have a drought occurring, xylem tension increases, cavitation events are happening, you get hydraulic failure, and at some point you reach a critical water status and you get a meristeme damage. So this is about, I would say, one of our main ideas we have nowadays about drought-induced mortality. It's really about dehydration of a plant. So it's not so much about carbon starvation anymore, but we really move more and more to what it really has to do with the hydraulics of the tree. And another mechanism, and it plays into the same role, is that we also more and more identify that embolisms play an important role, but often we see they are not so important during the drought event, but we know that the plants also after the drought cannot immediately recover. So we have done a study on Pinus terrestre, and we looked on drought and recovery, and we have seen that also 2 days directly after rewetting and also 1 month after rewetting, there's no loss. The percent loss of conductance remains the same. So they cannot reverse these embolisms, only if they would regrow new tissue. So I think this is also more and more of our standard knowledge that we have. And based on this, Bill Hammond has done this nice studies on Pinus tardea. He also showed that there is a probability of mortality. This is here for about 80% of percent loss of conductance. When you reach this threshold, about 50% of the population have then died, so they didn't recover after rewetting. So I think this is more and more coming to our mind that particularly these embolisms and everything are very important when we have this drought release. And what we also know, because when we say, okay, drought mortality is mainly related to hydraulics, then we also, there's this really nice framework published by Arendt in a paper, and we also know that basically the effects on when do we have cavitation, when do we have P50, they all have to do with losing water, and this will be more and more evaluated the higher or accelerated the higher the VPD, basically. So the time when you're on a safety margin will become shorter and shorter with a higher VPD because residual water loss and water loss at Gmin will increase. And also the critical time until you reach mortality will become shorter. So higher VPD is really accelerating this. And I wanted to add something on elevated CO2 and the effects on drought mortality. And this is again a problem of Mac and converting it to Windows computers. So you can only see the curves, but you don't see the nice dots that have been in there. So there have been really data points in there. You can believe me. And what we see, of course, we have a lower stomatal conductance under elevated C and a higher under ambient CO2. But if we increase VPD, the rate of the decline was totally the same. So this is for Aleppo pine seeds that have been taken from the Yatir forest, and we have looked into this, and there's no difference then in the response. And then we also looked into, again, there's something missing, but when we looked into the probability until certain thresholds are reached, like the carbon balance became negative, the stomates are closed, and the turgor loss point, we don't see any difference between ambient and elevated CO2 seedlings of Aleppo pine. And they reached the mortality at the same moment of drought. So the time until death was not changed between these species. Yeah, but what about drought-induced tree mortality trends? I mean, now we have heard a lot about physiology, and we have getting, I think, a better understanding that really water loss can induce mortality. But what about trends? So what do we know so far? And when we look into what has recently been published by Bill Hammond, he looked into tree mortality events related to drought, in particular hotter drought events, and you can see they are spread all over the globe. So in every forested biome, you find incidents of tree mortality. And these data are all from peer-reviewed studies, and they are not systematically. So he basically did a meta-analysis, went into these studies, and found this out. And really, you can clearly see that there's also a hotter drought fingerprint. So the mortality happened during warmer and during drier conditions as well. So now we are in a dryland climate, and if you look onto dryland tree mortality, also using his data, his database there, then we can see that this also happened in areas that we can, with an aridity index below 0.65, that there's also tree mortality occurring all over the place, and it's not really directly related with the intensity of the aridity. So the more blue, the aridity index is very dry, and the more yellow, it's less dry. So you can see it's just happening all over the place. I wanted to just zoom on into two examples very quickly. So one I found really interesting of the Western Sahel by Gonzalez et al., and the other one in Western Australia on eucalypts. So the first one, they did a Transact study, and they covered the area between the time between 1915 and 2002, and they found that about the tree density declined by 20%, and they did not attribute it to human impact so much, but really to a change in precipitation that was strongly decreasing in this area, while temperature was strongly increasing. So this is another study that shows us there's really something going on, and the same in a site in Western Australia, where they also did an intense study on eucalyptus vandu, and they found that with decreasing annual rainfall, there's a decline in the health of these eucalypt trees. And this is very nice, and it gives us some insight so that we know all too little water can really kill trees. But how much do we know really about this globally? I mean, how much do we know about the state? How is this changing? We know it's getting, in a lot of areas, drier and hotter. Yeah, and we know really surprisingly little, we have to admit. And one of the reasons, that's why we formed also this network. It's really time for this global tree mortality network. So at first we started as a UFO task force monitoring global tree mortality patterns and trends. And as a spin-off of this task force, we assigned the International Tree Mortality Network. So Henrik Hartmann, Bill Hammond, Cornelius Senf, myself, Tom Pugh, Rupert Seidel, Bernhard Schult, and Adriani. We are basically kind of the core team of trying to organize this. We have a nice webpage, we have a Twitter account, what you all do nowadays, but we have also these really nice seminars, and some people have maybe attended some of them. Yeah, but this is not all what we want to do. So what we really want to achieve, but at first I wanted to highlight, okay, we have really members from all over the globe, so you can easily become a member, just send us an email. What we really want to do is our ultimate aim, and it's of course a very big aim, and we don't know if we will ever reach this. I mean, the first thing, bring together the community. That's something that we are working on already, but the main goal is really to quantify trends in global tree mortality rates, attribute the causes, and predict tree mortality trends at some point. But now, at the moment, we are trying to work on the first aim, and yeah, this is our vision behind this. Okay, we observe everything, we put this all somehow in the database, and there will be models to working on this. We have detailed experiments and so on and so on. But so there's a big challenge. So we know that mortality assessments are basically not coherent. So we have one big data source that we could where we could have data on tree mortality would be ground-based measurements, mainly forest inventories, but there are also remote sensing data that could be used. But often there can be a global coverage, but often they are not detailed enough so to really track the mortality rate. So we just get a canopy mortality, while from inventories we could really get stem mortality out. So the first thing we did then, we were looking into what do we really need to know to understand that a tree is dying, basically. So when you look onto a forest inventory, often it's done to really assess timber stand biomass and so on, how much timber is there, but not if trees are dying. So we said, okay, canopy mortality is the first one. You just need satellite data. You have a change in land cover. The second is stem mortality. We need tracked trees. So we need trees have to be tracked, they have to be numbered, and when they are gone, that needs to be identified. And yeah, of course, we need to know if the tree is alive or if it's dead. But if we want to have a bit more data, if we want to really know how the forest structure and the carbon storage is changing, then we need, for instance, volume mortality. We need the track trees, we need the tree status, but also the diameter, the height, and the species and the allometry. So it really depends on what you need, but one of the first things when you do inventories, it would be really easy here if you would have these track trees. So then we also went to the community out and we made a survey and we only asked about metadata. So do you measure forests and how intensely do you measure them? How are they monitored? So the meteorological information. And we also looked into national inventories that we found. In total, we got more than 300 entries from more than 80 countries, and we covered all forested continents. The number of sampling sites reported were more than 800,000. So based on this, we made this map, and the more yellow the area, the more forest area of the total area that is forested is surveyed in this country. Some countries are gray because they have less % of forested area, and I looked it up. I think Israel is marked as gray in there. Dark gray means we didn't got any data. And what is interesting that more than 120,000 hectares of forest are intensively monitored, but that only means that it's 0.03% of the global forest area. And if you look into this plotted over time per continent, then you can see that the good news is really increasing the amount of forest area that is surveyed, but you can see, okay, there's a big difference between like Americas and Europe have a high percentage of forest area that is surveyed, while other countries, or other continents, sorry, have way less. Yeah, from here I come to my take-home message, that climate change type droughts really pose a risk and they can amplify tree mortality. And I think that elevated 2 might have a mitigating effect, but that is not very clear and might only have a very marginal role, particularly when you reach a point when stomates are closed. Elevated CO2 doesn't help the plant very much. Yeah, and we also really currently lack a coherent observation of tree mortality rates. And we think as a community that we need these coherent tree mortality metrics. We need to invest into ground-based data collection and also empower those that are collecting the data. That means really a fair policy, that there are a lot of people doing the hard work, but then they are not part of the really nice papers that might come out from that. And also integrate ground-based data from different networks and remote sensing. So thank you so much for this. And I hope you have a nice evening later too.
So we have Yael Wagner, she will talk about embolism again.
So I will talk about embolism, and I just want to touch base about the terms because I'm going to use several of them. So I'm going to refer cavitation as the instance when an air bubble enters the water conducting conduit, it expands and blocks it. And then the result is embolism, which is the actual state of the blockage. And as was stated before, when embolism happens on increasing rates, we can get to hydraulic failure, which is considered to be a major driver of drought-induced tree mortality. So let's move on to an assumption. Embolism is associated with hot and dry climatic conditions. We can probably unanimously agree about it. And why? Because when you have low water supply and you have and high atmospheric demand, you increase the tension between the water molecules, you decrease the water potential, and these induce cavitation and embolism buildup. But what are the actual evidence that we have to support this assumption that we can probably agree about? So for years now, we had methods to quantify embolism. Just to name a few, there's the cavitron, the Sperry apparatus, and even the microcity, which allows us to directly observe the embolism. But these all represent one point in time. They're not continuous. And if we cannot monitor cavitation continuously as it occurs, we cannot pinpoint the exact moment when they happen, and we cannot correlate it to the exact environmental conditions that induce this embolism. So we cannot make this assumption that we all agree upon empirical. In recent years, there has been the development of the optical visualization method sensors, or the OV sensors, and these sensors can detect the actual cavitation moment, so monitor the cavitation as they occur during drought. So to make this connection, we went to a Yatir forest, which already appeared here several times. You can see it in the satellite image. It's a very dry planted pine forest. The annual precipitation is very low. There is a very long, very dry summer. And in this forest, we have, again, the plot that was also mentioned here, established by Professor Dan Yakir, when we can get the instantaneous environmental conditions. So check. And in this plot, we installed the OV sensors on the mature trees in the field. So again, we can monitor continuously and compare the instantaneous environmental conditions. And to that, we added monthly checkups of microcity and water potential just to reaffirm the continuous measurements. So first thing first, could we detect cavitation? And the answer is yes. We detected over 200 cavitation events over the dry summer. You can see the buildup of embolism, or the percent of embolized pixel area in the kind of green lines here, from zero at the beginning of the dry season to about 12% in the end. And just to reassure you, these results matched pretty nicely the results of the micro-CT, both in dynamics and in values. So, could we make the correlation between hot and dry climatic conditions and cavitation? So let's break it down to the two parameters of drought that I mentioned, the atmospheric demand and the soil water content. Here you can see the spread of these 200 cavitation events along a scale of VPD gradient. And you can see a pretty dramatic increase as VPD increased, and it reached as high as 42% chances to get a cavitation event when VPD was higher than 5 kilopascal. And what about the soil? Well, we actually couldn't find a very clear relationship there. And these results sit pretty nicely with a recently published paper by Schoenbeck et al., where they experimentally manipulated VPD and soil water content, and they found that embolism can occur under high soil water content when VPD is high. So I showed you 12% embolism over a period of 5 months of dry summer. What is the physiological meaning? Is that a problem? So to look at that, we wanted to look at the climatic history of the plot. So this is a kind of a climatic matrix of the Yatir forest. On the x-axis, you can see the lowest soil water content of every year. And on the y-axis, we have the number of days with high VPD. And I labeled in red the year in which we measured the 2021, and you can see it was not an unusual year. So we can expect about 12%, maybe higher, on an annual basis in these trees. Well, if I can turn your attention to a year that was extreme, so this is 2015, up there in yellow, this year had over 60 days with high VPD. And the following year, 2016, there was an extensive mortality event. And when Preisler et al. Tried to trace the first signals for mortality, they found them in September 2015. So right when this very extreme summer was coming to an end, the trees already started to give mortality signals. And if we consider that heat waves are going to increase, we can expect higher and higher levels of embolism on an annual basis in these trees. So just to sum up, we were able, for the first time, to monitor or to observe cavitation in mature trees in the forest. Embolism reached about 12% at the end of the dry summer, and they were pretty closely linked to high VPD, not so much to soil water content in our case. And increasing heat waves and drought events can have fatal consequences for trees, possibly through embolism. And I just want to thank everybody who supported this research, and thank you for listening.
VPD are exceeding 5. When did it occur? Did you have in the spring and in the fall?
It mostly happens in the fall and spring, but specifically this year there was a very long heat wave in August.
Cumulative embolism, right? So you had that big jump in the beginning of August, right?
Yeah. Yeah. This one. Yeah. It was, I think, it was longer, but I think we had about 5 days of over 5, and then it was like maybe 4, maybe. So it was pretty long.
From someone who doesn't understand anything about plants. So cavitation is the physics of water. And why is embolism associated with cavitation directly? Can it happen with other physiological occurrences in the plant?
So the water contacting tissue is under tension. So if you have an air entry, it would expand because of the negative tension, and then it would block the conduit and create embolism. So if it would happen because an air was sucked into it, or because an animal took a bite, or a branch broke, is this what you mean?
I had a similar question, but I think you're physically inspired probably, because you can basically in plant science not call what we see cavitation. We in the plant physiology community we still sometimes use it, but we try to not use it anymore, because cavitation is a physical process where the air bubble spontaneously suddenly is there. And in plants, then for having that you have to have up to
It doesn't happen spontaneously. Yeah.
Is indeed what you say, this air entry, and therefore we cannot call it cavitation from a physical point of view, because you see that confuses
So I'm sorry, I wasn't aware. I thought there were like 3 mechanisms for cavitation, so the air seeding. Okay,
the
sorry.
air seeding is indeed, but it leads to embolism formation. So
Okay.
it's the embolism
Sorry.
that you see,
So I'm sorry, it's just a common term in plant physiology.
Some try to now point it out, but it's still not so common. So spread the word from now on.
I will.
And,
From now till the end of my PhD.
okay, good, excellent.
We have Timo Knuever, singer, right? Yeah. Okay. And he will speak about hydraulic modeling and legacy effects.
Thank you, and before I begin, I also would like to thank Uri for inviting me. Well, not actually me, but my colleague, Daniel Nadal-Salah, who couldn't make it here, and this is why I now have the opportunity to present his work. So I'm talking a little bit about modeling drought effects at the arid timberline. So, and to jump right in, we know that drought stress influences tree hydraulics and physiology. We know with climate change we have rising, increased frequency of severe drought periods, accompanied by rising temperature in VPD and less precipitation. This has various effects on plants and their physiology, like the reduction of stomatal conductance and transpiration and decreasing xylem water potential. This leads to less carbon uptake, depletion of water storage, loss of hydraulic conductance, and leaf shedding. Now to jump into the picture where we want to model this, we all heard about the Yatir forest, this plantation of Aleppo pines, which is an ecosystem that survives on very little water, so super interesting. We've heard a lot about that, and we've also have seen that before. So the net ecosystem exchange and evapotranspiration are tightly coupled to that small window of rainfall in the beginning of the year. So now our question is, can we capture these drought responses in a model? This is all work in progress by Daniel Nadal-Salah and Rüdiger Grote. And we before have used the landscape DNDC model, which is a process-based model, to capture forest structure, physiology, ecosystem, carbon and water fluxes, and soil processes to get drought responses. Now, how did that work out? Not so well, because we have two main challenges remaining with the standard LDNDC model, which is our VPD response was underestimated, and our drought response was overestimated. And then there's still something missing in the model, which is damage by drought, which obviously affects tree physiology. So how does our new model work? We started out with it's based on the standard SOX model presented in Eller et al. We have the soil water potential and soil water content, which defines the soil water potential, and with the amount of fine roots defines the root water potential. From there on, via the conductivity from the root to the canopy water potential is defined, and also the conductivity from root to canopy influences the stomatal conductance, which of course is influencing the net assimilation and the gross primary production, which has a feedback loop with the leaf area and also with the sapwood area. Leaf area and also stomatal conductance obviously influence the evapotranspiration of the canopy. So what we first included were the non-stomatal limitations, directly linking the canopy xylem water potential to the net assimilation, which was presented by Nadal-Salah in his developed SOX Plus model. And then we wanted to include damage and dehydration into the model, which plays a role here. So with loss of the conductivity in the root and the stem, from the root to the canopy, we have loss of sapwood, which directly then influences loss of leaf area, because less carbon is fixed and less leaf area will be produced. And also we have dehydration parameter in this model. This happens when the root and soil water potential is disconnected, and then the canopy water potential defines root water potential. So how did our model turn out to be? First, this is the soil water dynamics, and we can see in red our modeled data and in blue the observed data. And as we can see, the model pretty well fits the observed data. The same goes for transpiration, again in red the modeled and in blue the observed. Here the observed transpiration is derived from sap flux data, And this is our model transpiration, which quite well fits. And now to the tree water potential dynamics. Here we have in red the canopy water potential and in blue the root water potential, the modeled root water potential and modeled canopy water potential. And in black we have the observed water potential presented in Price et al. And the gray bar shows the difference between the root water potential and the canopy water potential. So the difference should be relatively small when there's a drought period and the stomates are closed by the plants, and we have a larger difference if there's not a drought period. And the maximum observed water potential refers to a maximum of 10 to 20% PLC. So what are limiting drivers for a cross-primary production? For that, we compared our modeled difference in water potential between canopy and root and the soil water content. And as we can see in summertime, when there is low water content available, there's SVC, so soil water content is limiting. And in winter time, we have a change and the VPD is limiting. Now coming to the drought damage, we modeled that we have always with dropping pre-dawn water potentials, we have a decline in sapwood area. This refers also to about 20% of the sapwood area. But then again, we can also see the regrowth in times when there's enough water available. And last but not least, the leaf senescence. So we again have observed in blue and simulated in red. And we defined two seasons, the phenological season, which basically is just a phenological leaf fall, and the drought-induced leaf fall in the stress period. And again, we can see that our model well caught this observed data. So what could we learn from our modeling? We have the new hydraulic implementation in the LDNDC model that captures the drought responses in the Yatia forest. We found the most limiting drivers that were identified with the hydraulic approach, so the VPD and the soil water content. We found that the hydraulic dysfunction is reflected in sapwood area and leaf area decline. So we have new ways to assess drought legacies and responses to future conditions, and we are able to simulate drought-induced tree mortality based on sapwood decline. So thank you all, and looking forward to questions.
Last and best talk of the day. Assaf Alon will speak about mortality of cupressus, right?
Yeah.
So now is the best time to lecture. That's why we kept it for this.
Okay, so I want to introduce my research, which is on the topic of drought-induced mortality for cupressus sempervirus, correspond with 50% of embolism. Cupressus sempervirus is the common name of it is cypress, and it's a kind of conifer that is grown also in Israel, and I will use the term cypress from now on. So we'll skip the introduction of plant in drought. So the background for my study is a debate that there is in the literature, which is what is the degree of embolism that plants can survive. So in the left we see a study from 2010 of Broadry-Betan et al., which worked on the species Calitris. And they report on a correlation between P50 and the water potential that causing mortality, which means that according to that, this species can, this is a family of species, but it means that they can survive up to 50% of embolism. And in the other hand, research from the recent years that I mentioned also before of Will Hammond, that work also in conifer but pine. And this shows that the P50 of this pine is minus 3 megapascal. And he shows that mortality is started from above 70% of embolism, and survival can be up to 90% of embolism. So this is a contradiction in the literature, what degree of embolism the plant can survive from in terms of the mortality. And my study was take place also both in field measurements and also in controlled experiments. So first of all, I did field measurements which took place in Israel in 3 different sites from the north to the south along the climate gradient, which 3 sites that represent different climates: Mesic Mediterranean in the north, Mediterranean in the center, and semi-arid in the south, which correspond to different amount of annual precipitation. And first of all, we also measured by Cavitron the PLC curves for the cypress from the different sites, and we saw that it was very highly resistant to embolism with P50, with values of P50 range from minus 10 to minus 13. And we also measured for 2 consecutive years in the dry season water potential pre-dawn and midday. And we see here the correlation between them. In the x-axis is the pre-dawn and the midday in the y-axis. And every point here is an individual measurement of individual, and the different colors represent the different sites accordingly to the colors. And we can see here that this plant is reaching its equilibrium point at around the values of minus 5 megapascal, and the minimum water potential is minus 6 megapascal, minimum water potential that was measured in the field in the 2 years. And something interesting that we saw, it's a few months later after the dry season, we saw that mortality of 2 of the individual in the semi-arid site. So this led us to the hypothesis that mortality of cupressus of cypress is correspond with less than 50% of embolism, maybe something similar that we see in the calitris. And to test that, we did a controlled pot experiment which took place in Vulcani. And I present here some of the results. We also worked on a comparison between 2 types of cupressus, but for this hypothesis, I present just this result of one of the types that is based on seed source. So it was done in summer 2021. So generally, we applied drought by closing the irrigation in some point after we grow the seedlings for a few months in the greenhouse. And then we stopped the irrigation and let the plant dehydrate. And in the meantime, we measured a lot of parameters, photosynthesis and water loss. We scaled the plants every 2 days or 1 day. And also we measured the water potential and relative water content. And we wanted to get to a certain threshold of water potential and then re-irrigate again, and then see if the plant is recovered. And because of that, we couldn't measure values less than minus 8 megapascal. We correlated all the way for all over the experiment, the relative water content and the water potential to predict, and we use it to predict values that are lower than minus 8 megapascal. So At the end of the experiment, we re-irrigate the plant, and we waited for at least 2 months to see if the plant is dead and if the canopy went to complete brown. So we decided that the plant is determined that the plant is dead. And if we take a look here, this is a mortality image for all individuals in the pot experiments. That the water potential here is the red and the green bars are values of water potential for the tree that was dead at the end of the experiment and the tree that recovered. And these values are based on the predicted water potential that we predict from the correlation between relative content. And we have also a few measurements of before minus 8 megapascal of pressure chamber of these individuals. So what we generally see here that we measure also for this type of compressors P50 by Cavitron. We see that from values of minus 10 megapascal and beyond, we see full mortality of compressors. Values of minus 8 and above, we see a full recovery. And in between minus 8 to minus 10, we see this is the risk zone, as some of the plants die and some of recover. We also measured at the day of re-irrigation electoral leakage of the leaves. And we see here that we see, first of all, a significant influence, significant difference between the relative, the electro leakage of the recovered and the dead plants. And if we correlated the relative water content, the electro leakage to water potential, we can see that there is a kind of threshold that above 30% of electro leakage, there is mortality. And it also corresponds with the P50 of the plant. So the conclusion from this study, from these results, are that cypress is controlled in controlled condition. First of all, it reached and recovered for lower water potential that the minimum water potential that he reached in the field. So this was surprising to us. Tree mortality in cypress is linked directly with water status parameters such as relative water content and water potential, which can define thresholds of no return. And electro leakage higher than 30% indicates mortality and corresponds with water potential lower than the P50. So thanks for your attention. I want to thank my advisors Rakefet David Schwartz and Shabtai Cohen, and all the people that helped and collaborate in this research and the funding.
Okay, it's dinner time.